Good afternoon. My name is Xiaolei Su. I'm a faculty at the Yale School of Medicine. Today, I'd like to share with you about my idea in developing a new version of CAR-T with improved antigen sensitivity. I'd like to start by introducing my team, which includes myself and my postdoc, Xinyan Zhang, student Fawzan Hashmi, and my collaborator, Sidi Chen, who pioneers in genome editing technology, and Iris Isufi, who is a clinician and the director for CAR-T therapy at Yale New Haven Hospital. So the challenge we're trying to address here is the current CARs have low antigen sensitivity, which means they can only target high antigen expressing cancer, but not low antigen expressing cancer. So the low antigen expressing cancer will escape the surveillance from the CAR-T. And for the high antigen expressing cancer, even they have the patient has very high initial response, like over 90%. But after 6 months, half of the patient will show a relapse. So basically the tumor grows back. And this is because tumor downregulates their antigen expression level below a threshold that can be detected by the CAR-T. To address this problem, we developed a called IDR CARs, which show the enhanced antigen sensitivity. And so now these IDR CARs can target low antigen expressing cancers. I think these IDR CARs will make a positive impact on cancer immunotherapy by expanding the target of CAR-T to these low antigen expressing cancer and also reduce the relapse frequency for these high antigen expressing cancer. So also I think this project presents a few innovations. First of all, these IDRs can be combined with CARs targeting a variety of different antigens, including CD19, CD22, or HER2. And these IDRs, because they are a protein peptide that can be fused to any CAR of your interest. So it's a pretty universal element. And second, these IDR CARs can induce biomolecular condensation of the CARs on the T cell surface, which increase the avidity of these CARs binding to antigen. So that's how it enhances its antigen sensitivity. And the biomolecular condensation right now is a really hot topic in biology. So we can learn a lot from the basic research, which will fuel the improvement and the design of new IDRs to improve these CAR-Ts. And last but not least, the sequence of these IDRs has nothing to do with CAR-T or T-cell signaling. So it serves as an orthogonal approach to improve the CAR-T and can be combined with other current approach, including introducing the co-receptor signaling or like cytokines to achieve additive or synergistic effect. Actually, there are hundreds of IDRs in the human proteome. Through some computational approach and wet lab, we can narrow down to these 3 IDRs that can robustly promote the condensation of the CAR on the T cell surface. And here I'm showing you that 2 of them, the IDR1 and 3, represented by the orange and the purple curve, that can significantly enhance of the killing of low antigen expressing cancer cells by the CAR-T, as shown on the left, and also it enhances the survival of mouse in a xenograft model, as shown on the right. Okay. With the support of this fund, I'd like to test the safety of these IDR CAR-Ts, because I think it's critical to make sure that these new IDR CAR-T does not induce substantial cytokine storm and tissue toxicity, which I think is a key issue to be addressed for all the CAR-T therapy. And the goal here is really to identify the IDRs that can achieve a balance between a good tumor eradication and the tissue toxicity. So with that, I'd like to thank all your attention. Happy to take any questions.
That was perfect. Question from the judge.
What mechanism does the IDR drive increase?
Yes, so what mechanism drives the increase the antigen sensitivity by IDR? So IDR is it stands for intrinsically disordered region. So it's a protein fragment that can promote self-assembly of CAR to form these condensates. So you can think about it's a clusters on the T cell surface. So that will increase the avidity of the CAR binding to the antigen. So basically, the traditional CAR only have like one interaction site with the antigen, but by form the condensation, you have multiple actually interaction sites. So that's how it can recognize the low antigen.
Awesome. Perfect. Thank you.
Thank you.